our next topic is the special senses. We're going to explore the 4 special senses. The 4 special senses are vision, or sight, and then hearing and equilibrium, joining together structures governed by what's happening within your ears, and then olfaction, which is the sense of smell, and then gustation, the sense of taste. We're going to spend most of our time on vision in this lecture, and then hearing in the following lecture. For the other 2 senses, olfaction and gustation, there's not a lot of specialization that's going on there, and it's not worth discussing in a whole lecture context. So just to cover these quickly so that we have them covered, each of these is governed by chemoreceptors, and those chemoreceptors are either in the nose for olfaction or in the tongue for gustation. And what happens with these is that you get a binding of the receptors or groups or set of receptors, and then that sends a signal or an action potential into the brain. And it is actually the combination of the receptors that produce the many different smells and the many different tastes that we can come up with. So in the nose, we see at the top of the nasal cavity the olfactory region, and within this olfactory region is where we have the specialized receptors. And if we take a closer look at those, we see that those are simply a series of receptors that are then bringing in action potentials right into the olfactory bulb and into the brain. So again, it's the binding of combinations of these receptors that will give us lots and lots of different smells. We know that in order to taste, we must also smell and do that very well. So we know when we have a plugged nose, for instance, you can't taste anything at all. So these are also important in gustation as well as olfaction. In looking at taste, we see the tongue covered with taste buds. And for the taste buds, you're going to have different regions, whether you're on the sides or at the tip or at the back, that are going to be for sweet, sour, bitter, and salty, the basic 4 different tastes that we can get. And then in addition to the tastes that are coming in terms of sweet, sour, bitter, and salty, you also have those olfactory signals coming in to give us the different tastes. Okay, so that covers the sense of smell and the sense of taste in a quick way. And again, not a lot of specialization there, so didn't want to spend a lot of time on that. What I do want to focus on is vision. For vision, we see that we are trying to take light rays and transform them into an action potential or an electrical impulse. And to do that is pretty impressive, but then to add on top of that we get this really dynamic color image that is constantly being refreshed and changed. So that formation of the color image, yes, but not only that, but also the ability to get it to refresh so quickly and give us a moving picture, a constant moving picture. Okay, so overall the functions of the eye are going to be to bring the light in, so to collect the light rays, and then we want those to be carefully focused onto the retina. So we need to bring in the light rays and then focus them on the retina. The first thing that we'll start with the eye is the structure of the eye and the eye wall. We see 3 layers to the outer eye wall. The fibrous tunic. The fibrous tunic is made up of the sclera, that's the white, and then as you come around to the front, that layer becomes clear, and that's the cornea. The second layer, which is filled with blood vessels, the vascular layer, will be composed of at the back of the choroid coat, and then as we move around to the front, we have the iris, and that is, if you have blue eyes, the iris is blue, that smooth muscle is blue, as opposed to if you have brown eyes, then that muscle is brown. The sensory tunic is the retina, And the retina is composed of the sensory layer, which we will see and look at the details of the neurons that are there. And then also there is this pigmented layer, which is basically going to be a black or dark background that we're going to form images against to make visual acuity much better. So here, looking at the eye, again, we see the sclera within the fibrous tunic. And again, as you come around to the front, that sclera becomes the cornea, and instead of it being white, it is then clear. Inside of the fibrous tunic or the fibrous layer, we see the vascular tunic, and the vascular tunic at the back is the choroid. So this region is the choroid coat, and it is filled with blood vessels. And then as we come around to the front, the choroid leads into the muscle, the iris. So here we can see the iris muscles on the front side. The innermost layer, the nervous tunic or the sensory tunic or the sensory layer, is at the back, the retina. So here we have the retina lining the back. That's where we're forming the image, and collecting those light rays and transforming them into action potentials. And then there is no counterpart to that as we move around to the front. We have a lens. The lens is for focusing. So anytime we get light rays coming in and they hit a fluid or they hit the lens, they're going to bend, and that is called refraction. So refraction is simply the bending of light. So the light rays come in, they pass through the lens, they pass through some other fluids that we're going to discuss in a moment, and they form an image on the retina. So you can see if light rays are coming in over here, they're going to hit the retina up there. Light rays coming in over here, they're going to hit down here. Light rays coming straight in are going to go straight back. Okay, so the lens, that's a growing structure, growing living structure. There are two layers of cells, one layer of cells facing towards the front, one layer facing towards the back, and that lens is going to serve as the division between two different areas within the eye. So as you can see here, we have these two areas listed right here that are called the anterior and the posterior chamber. We don't want to use that word chamber, and it is to confuse what I'm going to talk about next. So when I refer to two different areas within the eye, we see the lens, and then we see the area behind the lens, and we will refer to this as the posterior compartment. And then we have the area in front of the lens, and that is the anterior compartment. The anterior compartment is going to contain a fluid called the aqueous humor. In the posterior compartment, we find a different fluid, and that is the vitreous humor. So we're calling this the vitreous chamber. We want to call that our posterior compartment. And where they call this the anterior and the posterior chamber, we want to call that the anterior compartment. So those fluids that are within the anterior compartment and the posterior compartment are very, very different. The fluid that is in the front is referred to as the aqueous humor, and again, that is a very liquid state fluid. It is highly fluid. It is like water. Not like water in its properties or in its contents, but like water in terms of the way it behaves and the way it flows. In the back, though, we have the vitreous humor, and the vitreous humor is a solid jelly-like mass. And so the difference between these two fluids is that this one is like jelly, and this one is like water. The jelly-like fluid, the vitreous humor, is the same vitreous humor that you've had for very, very long. If I had some way to go in there and mark that fluid, what I would see is that it's not changing. It's the same fluid every single day. If I look in the aqueous humor and I were to put some markers in there, what I would see is they would be cleared in a short time. So we see that the aqueous humor is being cycled. If you've ever gone into a doctor's office for an eye exam, they'll tell you to rest your chin on the strap and then pop. They hit you in the eye with a puff of air. So that puff of air is hitting the surface of the eye and looking at the tension. So if this fluid, the aqueous humor, is being cycled, that means there's a potential for it to be not cycled out quick enough. That would increase the pressure inside of here and increase the pressure overall inside of the eye, and that could lead to glaucoma. What happens in glaucoma is these tiny arteries would not be able to deliver all of the blood all the way to the forward part of the retina, and we would start getting blackouts in these areas, and you would develop tunnel vision. So the aqueous humor must be cycled. The vitreous humor, though, the same vitreous humor you have today is that tomorrow and the next day and the next day. It will help in or aid in interocular pressure, just like the aqueous humor. They're both involved in pressure within the eye, but the vitreous humor is not going to change. The aqueous humor is the one that is cycled and the one that will change. It needs to cycle because if we remember back that the iris is where those blood vessels are. So there's no blood vessels lining right underneath the cornea like we have the blood vessels lining right underneath the sclera. So in order to feed the nutrients to the cornea, we have to definitely have the aqueous humor cycled as a blood filtrate so that it can provide the nutrients. So glucose-like molecules would be dissolved into the aqueous humor so we can deliver those to the cornea. An accessory structure to the eye is also shown here, and that is the conjunctiva. The conjunctiva is a transparent membrane that lines the cornea, starts to line the sclera, and then it would wrap back and line the inside of the eyelid. So it follows the cornea, goes over to the sclera, and then it would line the inside of the eyelid as well. So it would form a little pouch down in this region and a double layer, because one layer is covering the eyelid and the other layer is covering the sclera and the cornea. That little pocket has the ability to trap some bacteria every now and then, which would cause swelling. And swelling, you change the word at the end to itis, like appendicitis. You take your appendix, add itis, means you have an inflammation of the appendix. Well, an inflammation within the conjunctiva is conjunctivitis, otherwise known as pink eye. A very communicable disease, easy to pass on, because if those bacteria can find this nice warm and fluid-filled space within your eye, you touch your eye, you pass it on to the next guy, and boom, they have the same problem within their eye. Okay, well, next let's look at the accessory structures of the eye. To be an accessory structure of the eye, it must be something that will protect, move, aid in eye function. And so protecting and moving, well, we see eyebrows as protection, we see eyelids as protection, we see eyelashes as protection, we even see the conjunctiva as protection. The lacrimal apparatus is going to be involved in moistening the eye. So protect, lubricate, move, and aid in eye function. These are definitely, the lacrimal apparatus is going to be involved in the lubricating and moistening of the eye. And then move, we definitely have the extrinsic eye muscles. So let's look at all of these a little bit closer. So first of all, we have the eyebrows. And the eyebrows are not necessarily these hairs that we see here, but more the ridge of the brow line, and that is, it is going to cast a small shadow over the eye and make it so that you look from the dark to the light. So looking from the dark to the light is going to aid with your visual acuity. Some people say protection also, because you're not going to have sweat pouring down off of the forehead into the eyes. Okay, I would buy that as well. Then we have the eyelashes and the eyelid. The eyelid, movable protective coverings that are blinking up to 25 times per minute. And again, that's just to spread the tears across the eye. The eyelashes, catching larger dust particles, not allowing those to fall into the eye. So a protective mechanism there. We also have, again, that conjunctiva. The conjunctiva would be covering the surface of the eye, again, as a protective layer over the cornea, protecting that cornea. And then we also have the lacrimal apparatus. And before we move to the lacrimal apparatus, we'll notice that it is going to be positioned right at the outer upper margin of each eye. It is going to allow for tears to be released. Those tears will move across, and then those tears will drain into the nose. So we get a little different picture here. Here's that lacrimal gland, the lacrimal ducts, the tears being released, and the tears go into these little drains. The little drains are referred to as puncta, and we drain into the lacrimal canaliculi and into the lacrimal sac, down into the nasolacrimal duct, and finally into the nose. We know that the tear gland system, the lacrimal apparatus, is tied into the nose because when we get an overproduction of tears, we see that we get the sniffles also. The lacrimal gland is tied into the limbic system, Limbic, and the limbic system is your emotional center. So whether you're really, really happy or really, really sad, you could get tears being released from the lacrimal gland and overproduction of those tears, so much so that you get more than can be picked up in the little drains, and you'll get teardrops falling down the face. Finally, we have the eye muscles. And the eye muscles, if you think about an eye and you were to say, well, how many muscles would I need to move the eye? Well, you'd want to move the eye up and down. You'd want to move the eye left and right. So you would think 4 muscles ought to do it. Well, you also have to have the ability to roll your eyes and look at different points in between the up, down, left, and right. And so we also have 2 additional muscles, and those are the superior oblique as well as the inferior oblique. Those are attached at an angle, so an angled muscle is referred to as an oblique muscle. The superior oblique attached at the top, the inferior oblique rounded around to the bottom. And so we see that we have these 2 oblique muscles, and then we do have the 4 straight muscles. Any of the straight muscles is referred to as a rectus muscle. So the one that's attached at the top of the eye, the superior rectus, the one that is along the outside of the eye, so we're looking at this angle, so out here is the lateral rectus. The one right next to the nose would be the medial rectus, so here's the medial rectus, and then we have to see the underside of the eye here to see the inferior rectus. So if you were to contract both of your superior recti, that would cause you to look up. Contracting both of the inferior recti would cause you to look down. Contracting both of your medial recti would mean that you cross your eyes. And contracting both of your lateral recti wouldn't work. Something like a lizard or a duck would have independent control of those eyes, but we are unable to contract both of our lateral recti at the same time. To look right, you would contract one of the lateral recti and one of the medial recti to look right, and then you'd switch to contract the other lateral recti and the other medial recti to look left. All right, let's explore the retina a little bit. In looking at the retina, we have the sensory layer, or the sensory retina, composed of 3 different sets of neurons. The photoreceptor neurons, these are the ones that are going to transform the light rays into action potentials, and we will see that these are made up of the rods and the cones. And then we'll see a layer of neurons that's just transferring information. So from one end to the next, just bring the information along, and those are the bipolar neurons. And then finally we have the ganglionic neurons, which start with their cell bodies all clustered up, thus the word ganglion, and then we get them leading into the optic nerve. At the back, behind the sensory retina, we will see the pigmented retina, and this is that pigmented layer that's going to help us with visual acuity and the ability to trap light and not let it just bounce around the inside of the eye. So a nice black background to form an image against. So let's start off and look at that black dark layer, the pigmented retina. It's right up against our choroid layer with all of the blood vessels. Okay, so then we have the pigmented layer, all these black cells, and then here's the rods, and then here are the cones. So rods and cones, those are the photoreceptor neurons. Photoreceptor layer is where the rods and the cones are located. Then we have the area that's just a transfer center. That's our bipolar layer. And then finally we get to the ganglionic layer, which all of the neurons of the ganglionic layer are making up the optic nerve. Now, as we see this system, it's a little bit troubling for me because we can see here that we have our light source and light is moving this direction. We then see over here it tells us about action potentials, and it tells us that our action potential is going to move in this direction. That's a little bit troubling because if you think of neurons, photoreceptor neurons, bipolar neurons, and ganglionic neurons as wires, what you see is the light passes through all of these wires and then is transformed into the action potential, and now my action potential is moving this way, but that means that I'm moving towards the middle of the eye, and I'm going to bundle all of those neurons in the center of the eye, and then I got to bring them outside of the eye, which means I'm going to have to poke a hole in this system and lead the optic nerve out, which is going to leave me with a blind spot. I'm going to have a place where I can't have rods and cones at all. They'll be absent right there, and that will be our blind spot. So that's a little bit troubling about the arrangement of the eye, but we're going to see how we can get past this. We're able to get past this blind spot because we have two eyes. So binocular vision lets one eye look into the blind spot of each of the other eyes. So the right eye looks into the blind spot of the left eye, and the left eye looks into the blind spot of the right eye. Now, your next obvious question is, well, what if I close my eye? So if you close one eye and you look out, you don't see a blind spot there either. The blind spot is in the peripheral vision. And so what your brain is doing is your brain is filling in that gap so that it's not confusing to you. We wouldn't want a large black spot following us around all the time. So the blind spot is, if you look straight ahead and you look at, maybe there's a white wall off to the left of your vision or the right of your vision. And what you would see is white wall, white wall, white wall, and then a gap, and then white wall, white wall, white wall beyond that. And so what your brain does is it just says, well, if there's white wall on that side and white wall on that side, fill in the white wall between, and that won't confuse the vision or cause them to constantly be trying to look over into that blind spot. So the brain covers that portion of the vision that is not able to happen in the blind spot. And then in addition to that, we have binocular vision covering us for the ability to see in the blind spots. So before we get there, though, let's look at the rods, and then we'll come back and look at the cones. If we look at the rods, we see them as a column of a membrane that is folded back and forth, back and forth, back and forth. And within that membrane, so here's a lipid bilayer, phospholipid bilayer, and embedded within that membrane we see a molecule, and that molecule is called rhodopsin. The rhodopsin molecule is made up of two different parts, and that is the overall larger protein is referred to as opsin, and then there is a smaller protein embedded in that, and that's called retinol. And retinol is related to vitamin A, and thus people who have a good source of vitamin A, and people say you should have that so you have good vision, this is why, because retinol is a protein within the rhodopsin overall protein. So looking at this rhodopsin molecule, it is going to be affected by light. So in the dark, we will build rhodopsin molecules, and then light will trigger them to open. So this system is a little bit different in that we also have a gated sodium channel, and that gated sodium channel is being held open. So that's going to change our normal resting membrane potential. Our normal resting membrane potential of minus 70 now has an influence of a gated sodium channel being held open. With a gated sodium channel being held open, sodium is going to be moving into the cell, and so we shouldn't expect to be at minus 70 anymore. We should expect to be higher. And as we look at that being higher, what we would expect is that instead of being at minus 70, maybe we're all the way up to something closer to minus 30. So we're sitting at minus 30 instead of minus 70. That's the key within the I and the movement of an action potential. We're going to start at minus 30 and we're going to move from there. Okay, so the way the rhodopsin molecule is going to work is a pretty complex system. It involves some G proteins and phosphodiesterase. We're not going to worry about those details. All we're going to worry about is the rhodopsin molecule in the closed state is having no influence on the gated sodium channel. When it is hit by light and opened, that causes the gated sodium channel to close. Let's explore the rhodopsin cycle a little closer. In looking at the rhodopsin cycle, here we see the closed position, the dark configuration for the rhodopsin molecule, where the opsin protein has us in the closed position. Again, the sodium channel is open. When light hits the rhodopsin molecule, that causes the opsin, or the rhodopsin molecule entirely, to open, and then we get a series of events which causes the sodium channel to close. That sends the signal, that causes the action potential. We send the action potential, and it goes to the brain, and it says, you saw light. Then what we need to do is we need to recover. So the opsin protein is replaced within the rhodopsin molecule, and it comes in, and we're back to the closed position. The sodium channel is closed, but now we move back to the open condition, and we're ready for our next light impulse to come in. With our rhodopsin molecule closed and our sodium channel open, light hits it, causing the rhodopsin molecule to open and the sodium channel to close. And then we re-establish it just like we did before and get us back to the resting state. So if we were in really, really, really dark situations, we'd move all of our rhodopsin molecules to this. And as light hits, we would open it up and then cause the action potentials, saying that you see light. So the action potential is different. The action potential is you're sitting at minus 30, the light comes in and closes the sodium channel, and so you start to go back towards minus 70. You only get down to minus 35, and then we reopen this, and you go back to the minus 30. So this little hyperpolarization is the only thing we get for an action potential within vision. And again, it's caused by the rhodopsin molecule opening, and that triggers the closing of the sodium channel. So we've looked at the rhodopsin cycle as a way to adapt to two different light levels, but there is also another way, and it's a much quicker way, and that is pupil size regulation. We all know that if you look into a bright light, your pupil will constrict and limit the amount of light that comes in, and if you are in dark conditions, the pupil widens as much as it can to allow in any amount of light. So that will happen very quickly, and then the rhodopsin cycle will adjust to light levels over longer periods of time. So if you were in darkness for an hour, you would establish night vision, and that's because of the rhodopsin cycle. Eventually you would get to the point where you'd have so many of the rhodopsin molecules that they would start being triggered open by the faintest glimmers of light, and you would be able to see differences between lighter and darker objects in the dark. If somebody came in and turned on a bright light, or came into a dark room and turned on the lights, it would be painful because all of those rhodopsin molecules would be firing off at once, sending their action potential. And you'd have to shield your eyes. You'd feel like you were being blinded by the light. Okay, so that's the rods where we see the membrane folded back and forth and we see the rhodopsin molecule. The rhodopsin molecules would be all over in here, so each one of these rods would have many, many, many copies of that rhodopsin molecule. When we look at the cones, we see a tapered column instead of a straight up column. And as we look close, we see again that folded membrane, and we're going to see rhodopsin like molecules. Instead of it being a rhodopsin molecule, it's called iodopsin. So it's just IO and then DOPSIN. So the iodopsin has 3 different opsin proteins, and they still have the retinol involved in them, but the 3 different opsin proteins are responsive to different wavelengths of light. So what we see is there is a group of cones that are responsive within the blue wavelengths of light, a different set that are within the green wavelengths, and yet a different set that are within the red wavelengths. And so if I look at this particular shade of green, there's some small amount of blue cones firing off, a little bit more of the red cones opening up, and then we see many, many of these green cones. And so these green cones, these red cones, and these blue cones give us this color of green. And the yellow in here with this percentage of green and this percentage of red gives us this yellow color. So depending upon where you are in this color scale or scheme, we see different wavelengths of light triggering different amounts of the red, green, and blue cones firing open and sending their action potentials. The next thing I want to discuss is an accommodation to distance. That is, as an object gets closer, it is harder to see because we have to bend the lens more and more. So changing the lens shape to see things that are very close gets more difficult as we age. And the reason that this is because the lens is a living structure. And as I said before, it's made up of 2 layers of cells, 1 layer facing forward, 1 layer facing backward. And over time, those cells are going to get a little bit thicker and a little bit thicker and a little bit thicker. So the muscles don't weaken, the lens becomes thicker and it's harder to bend. So here we see this little gap represents that this object is very far away. So if we have a very far away object, when we hit the lens, we don't have to bend the lens much at all to bring that object into focus. It's only going to occupy this very small amount of space on the retina. But if we have something that's very close to the eye, those light rays have to be bent dramatically to get those images perfectly into focus on the retina. So the closer an object, the thicker that lens must become in order to get it focused onto the retina. So you might have recollections of a parent or a grandparent who you would think they need longer arms because they're holding that newspaper so far away from them as they're trying to pick up those small newsprint and focus them onto the eye. So they move them far away so that they can get them focused because the inability to make that lens so thick and bend it is being lost because that lens is becoming thicker and thicker with time. Here I mostly want to key in on diagram C here, and that is when we look at an object, it's projected onto the eye in reverse or upside down. And we already know this, that is the way that lenses work because that's the lenses that you've already dealt with in lab or some other time in your life when you used a microscope. A microscope inverts the image and makes it move left when you think it's moving right. And so the eye gets this inverted image. And so why is it that we see it right side up? The reason that we see it right side up is because of the brain. So the brain is involved in flipping the image for us. There was a set of experiments that was done that took a pair of glasses, and we put the glasses onto individuals, and that put a lens in front of this lens, which means that you'd see the telephone pole, It would get flipped over by the lens, it'd get flipped over by your lens, and it would be a proper projection on your retina. And in those experiments, what happened was the individuals were able to accommodate for that over time. In that, terrible headaches and terrible situations for them, but later, still those glasses on, the image projected right side up onto the retina, they would see those images as right side up in real life. If they took off those glasses, they would have the normal situation that we have here, and we would be seeing things upside down. So a set of glasses that you would not want to lose. That was reversed very quickly, though. Once you don't wear those glasses, you revert back to relying on this normal way things work pretty quickly. Okay, well, we all know people who have trouble with their eyes, either being able to see things that are close and then not see things that are far away, or see things that are far away and not being able to see things that are close. The condition where you are looking at something in the distance and can't make it focus means that you are nearsighted. You have the ability to see things that are near. That condition is called myopia. And quite often with myopia, the eyeball might be a little bit oblong so that it's longer from front to back. The normal light rays are coming in, they are bending through the lens, and they are coming to a crisp focus in front of the retina. So here's the retinal surface, here's where that focal point is, the nice clear crisp image is here, the retina is here. By the time these light rays were to get back here, they would have scattered, and you have a blurry vision. To correct that, a concave lens would be used. And that concave lens is going to bend the light rays out a little bit so that when they hit the lens and they're bent in, but the nice crisp focus is happening right at the retina now. So myopia is the medical name for nearsightedness, and a concave lens corrects myopia. If we look down at this condition, the eyeball is a little bit squashed from front to back, so that when our light rays come in and they bend through the lens, we get them to focus, but they're not focused here. They're not focused at the retina, but they are focused behind the retina. So here we have a blurry image. This is a farsighted condition, and in the farsighted condition, what we see is that is referred to as hyperopia. So hyperopia is farsightedness, and a convex lens will correct hyperopia. The convex lens is going to bend the light rays in a little bit. The lens will bend it the rest of the way so that we are a nice crisp focus right at the level of the retina. So myopia, nearsightedness; hyperopia, farsightedness. The concave lens fix myopia, the convex lens fix hyperopia.